Welcome back to the results section. As you can remember, our first research question was whether frugivores spatially aggregate their seeds, and if they do, at what spatial scale. We did this analysis, the cluster analysis, in the 3 study sites, and in the first study site that you can see, what we found is that the best model that fit our data was the simple cluster model. We also tried to fit other models, such as the complete spatial randomness, the heterogeneous Poisson, the inhomogeneous Poisson, or the double cluster processes, but these fits were put to the data. However, even the simple cluster model was not totally satisfactory, especially at small spatial scales, about 10 meters. For example, the distance to the nearest neighbor were larger than predicted by the simple cluster model for scales between 10 and 80 meters. Also, the existing gaps without fecal feces were on average smaller than expected. This spatial pattern of fecal distribution were largely impeded by the fecal marker behavior of buyers, which were particularly active or abundant in this area and made large latrines that they reuse frequently. So there exists some large cluster of fecal samples with string aggregation at very small spatial scales. In the linear-dominated side, the double cluster was the best model for the fecal sample distribution. Other models provided no satisfactory fits. The double cluster pattern was characterized by a strong aggregation at a small scale, from 1 to 10 meters, and a less marked clustering at larger scales, from 11 to 150 meters. Nonetheless, the observed clustering at smaller scales is even stronger than the one predicted by the model. The gaps without fecal samples were well predicted by the double cluster process. This also could be related to the presence of some Bayer feces in the area. In the Uniprus-dominated shrubland, the double cluster was also the best model for the fecal sample distribution in the area. Other models provided no satisfactory fit. This double cluster pattern was characterized by a strong aggregation at small spatial scales and a less marked clustering at larger scales. Also, the mean distance to the first nearest neighbor was satisfactorily predicted, as you can see. The observed clustering at all scales fit rather well the predicted one. Also, the mean gaps without fecal samples was satisfactorily predicted by the double cluster process. Interestingly, this site where we found the better fit, it is also the area with least Bayer activity, as you can see. In the second objective will be discussed in the next video. See you there!